bunch of different names. You'll also see synaptic knob and synaptic end bulb. So these three terms all refer to the same thing. It's the end of the neuron where the neurotransmitters are released. Okay, so it's this that we saw with the neuromuscular junction, right? It's got vesicles in it. Okay, this is ultimately where the signal's going to go to release neurotransmitters and then have our effect at the synapse. I think axon terminal is more of a current term. Synaptic end bulb or synaptic knob is an older term, but you'll still see it probably in different places.
Do we need to have a period on?
No, don't worry about that.
I was ready to just
Yeah.
show up to work this morning.
Yep, just dendrite, cell body, axon, and axon terminal, and you're good to go. And remember, if we're going to myelinate part of this neuron, what are we going to myelinate? What part do we myelinate? The axon, right? Increase that speed. Okay. And we talked about the axon hillock before also, and it's in that other picture. Axon hillock. Just make a note of that term now, and it'll make a lot more sense when we get to see what it actually does. Okay. So now we move down to the synapse, and we see that presynaptic cell or membrane, the cleft, and then the postsynaptic cell. And that's how we'll refer to things with the nervous system. Okay. So if you have a synapse, like with a neuromuscular junction, we had a synapse. It was just between a neuron and a muscle cell. So in our neuromuscular junction, we had a presynaptic cell, which was the cell that released acetylcholine, and then our postsynaptic cell was just the muscle cell, was the sarcolemma. Okay? So remember, the presynaptic is what releases the neurotransmitter. The postsynaptic membrane or cell is what has the receptors on it. Our structural classification, these you don't have to worry about, but you'll see descriptions of these. I don't know if you'll see pictures of these, but you might. And we'll highlight when we have some of these neurons. We've seen two of them already. These bipolar neurons, bipolar because there are two extensions off of the cell body. Right, so here's the cell body. We have a dendrite this way and an axon this way. Okay, and the dendrites branch up here, and then the axon, the same thing. Unipolar looks kind of weird, but with a unipolar, we usually draw it like this. So here's the axon. You see the axon is long, so the dendrites are down there, and then the axon is this whole thing. So this is a really long transmitting neuron with the cell body coming off to the side. We saw these somewhere in lab. Where did we see these kind of neurons? Remember? There's a collection of the cell bodies in the dorsal root ganglia. Our sensory neurons are like this. Okay? And we'll see why that's the case. And then multipolar neurons here with a whole bunch of extensions. Most of the neurons, I don't know what percent, probably 80% of all the neurons are this type. Okay. Bipolar neurons are really pretty rare. We'll see them in the retina in a couple places. All sensory neurons are unipolar neurons, okay? And we'll see why that's the case. So when we get down to the functional classification, we see the sensory neurons here, somatic sensory and visceral sensory. And then receptors, And the way the receptors are organized in this case are by interoceptors, exteroceptors, and proprioceptors. And with this classification, interoceptors are receptors that are monitoring things inside the body, internal conditions. So this could be things, conditions that we're not aware of. Most of them we're probably not aware of. Blood pressure, pH of the blood glucose level, anything you can think of that's happening inside the body. Some of the stuff we're aware of, right? You can pull a muscle and have pain inside the body, or the digestive system is a really weird system in terms of sensory stuff, because it's an internal organ that's controlled completely by the autonomic nervous system, but we get sensation from it, which we don't from most other organs. We do from the bladder, but not from the kidney. So if you feel nauseous inside, that's information coming from inside your body. So the signal that's sent is by an interoceptor, even though you're aware of it. Okay. And then extero, as you might think, these monitor everything external conditions. So these are all found, for the most part, on the surface of the body. So our special senses count as these. They all tell us something about the external world. Sense of touch, we have thermal receptors. So we have thermal receptors in both cases. Your hypothalamus has thermal receptors in it that monitor the condition of the blood inside the body. Exteroceptors monitor the temperature of the ambient temperature of the room, touch, pain, all that stuff. And the only one that's kind of weird is proprioception. And this is separated out because proprioception is the monitoring of your body position. So this is kind of a weird combination of both of these. And we have these receptors in all of our joints and skeletal muscles. And your cerebellum, that great big hunky part down on the bottom of the brain, it's got about half the neurons of the brain in it. Your cerebellum is largely responsible for this. Okay, so your cerebral cortex, the conscious part of your brain, determines where you want your body to be. You want to sit in a chair, you want to catch a baseball, you want to do something, but then it's up to the cerebellum and some other things to move your body into that position. And so it's a really complicated thing if you think about it. The cerebellum is monitoring the angle and the movement of every single joint in your body. So when you move your thumb, there's a receptor in the joint of your thumb that tells your cerebellum you've just moved your thumb. And then your cerebellum is going to say, does that match up with what I intended to do or not? And so if that is off, you'll see people, you can have neurological damage or degenerative problems where proprioception is off. And people will try to sit in a chair and they'll miss the chair because they want to sit in the chair and they see it, but their body doesn't do what they want it to do. Or I've seen videos of people who can't eat because they can't make it to their mouth. So when they get a fork full of food, they miss their mouth because their proprioception doesn't work quite right. And they can do, when they do neurological tests, you should be able to, like with your eyes closed, touch your nose and stuff like that. That's all a neurological test that tests some of this sort of stuff. It's kind of strange. Motor neurons, we don't need to worry about. We've seen those. Interneurons, what we're going to see is that in the movement of information in the nervous system, like this. Neurons that bring stuff in are sensory, right? Afferent. Neurons that bring things out are motor. They're efferent. But in between, when the signal comes in here and the signal goes out, there are potentially a whole bunch of neurons in the central nervous system that are being active. Those are all interneurons. So anything in between the sensory and the motor is an interneuron. In some cases, there could be a whole bunch of them. So your sensory information comes in, and then there's a neuron that takes it up somewhere, and synapses with another neuron that takes it somewhere else, and it goes bing, bang, boom, all back and forth here until it comes back down. So it's possible to have hundreds of thousands or millions of interneurons in the processing of stuff before your motor output comes out. It's also possible to have none, to have no interneurons in a reflex. Like a really fast, super fast reflex, the sensory neuron comes in like this and synapses immediately with the motor neuron that goes out. And so this is the fastest reflex you can have because you're contracting your muscle instantly. There are no interneurons in here. Okay? So the number of them will determine how quickly we can get that motor output out. Okay. Neuroglia, the next, let's see, after that, the next picture that I've given you, has all the information you need in it about neuroglia. So you can use this as part of your notes. And I'm going to highlight some stuff up here. So neuroglia are the helper cells in the nervous system. They do not generate an action potential. They compose a large percent of the volume. I have it in here. I don't, but they outnumber neurons about 50 to 1. So there are tons of them. Most of them are kind of small. Some of them are in the central nervous system only, and some of them are in the peripheral nervous system. And you want to know a little bit about each of them. Ependymal cells, these are the ones that we might be able to identify in lab. Okay. Key thing about these, assists in producing and circulating cerebrospinal fluid. You'll notice that they line the ventricles, those brain structures we talked about, and the central canal that goes down the spinal cord. So this cerebrospinal fluid, we'll get more details of it when we get to the brain, but since we're using that term, what we want to know is that the central nervous system is a really super delicate place in terms of physical structures and also in terms of the chemical environment. So your body's very particular about what can get to the brain and the spinal cord and what cannot. And so we have this structure here that we're going to see as part of the job of the astrocytes, this blood-brain barrier. This blood-brain barrier is formed by these cells that wrap themselves around capillaries. So remember we talked about earlier on that if you have your heart, you have an artery that takes stuff to a capillary, and then after the capillary you have a vein that brings it back. Right? This capillary is where everything's going to be exchanged with whatever tissue you're going to. So you have arteries that deliver blood to capillaries in every organ that you've got. All your muscles, all your kidneys, both your kidneys, everything. So the question when we get to a specific organ is, how leaky do you want this capillary to be? How much stuff do you want to get out of the blood and to go into the tissue? And we can make them more or less leaky. And so what happens is these astrocytes form around the capillary. So if this is a capillary, the astrocytes form around it like this. So now, what gets out of the blood and into the brain is determined by those cells. So that's the blood-brain barrier. And so we've got all the blood over here, and those astrocytes are only going to let certain things through to get to the central nervous system. And that's what becomes the cerebrospinal fluid. Red blood cells do not pass through that barrier. So your cerebrospinal fluid is not red, it's yellow. It's like plasma, sort of. And we'll talk more about what gets through there and what doesn't. Okay. A lot of drugs cannot get through there. Unfortunately, a lot of helpful drugs can't get through there, like antibiotics, which is why if you get meningitis or some sort of a bacterial infection in your brain or spinal cord, it's very difficult to treat. But anything that gives you a psychotropic effect, like alcohol, if it makes you high or makes you drunk, then it gets through here. And it's strange that so many drugs that do that get through there, right? Cocaine, heroin, marijuana, alcohol, all of it goes right through there. Just kind of strange. Okay. Cerebrospinal fluid. So astrocytes maintain the blood-brain barrier. Okay. And then all sorts of support. They regulate ion, nutrients, gases. Okay. Very important for astrocytes. The third really important thing that they do is absorb and recycle neurotransmitters. Remember that in our synapse, synapses all have pretty much the same type of a structure, where you have a presynaptic cell and then some postsynaptic membrane with receptors on it, neurotransmitters out here. It's always going to be the case that if we want the effect on this cell to stop, you have to get rid of these neurotransmitters. So we saw with acetylcholine, it's an enzyme that degrades it, right? In this case, what we're saying is there's a cell, there's an astrocyte cell out here, and that astrocyte cell sucks that neurotransmitter in to get rid of it. Okay? That's a process called uptake. So we can get rid of a neurotransmitter by enzymatically degrading it. We can get rid of a neurotransmitter by uptake here. And the third, you can write in the side of your notes, is we can also take that neurotransmitter back into the cell that released it. And that's called reuptake. Those are the three ways we get rid of neurotransmitters. If you've heard of Zoloft and Prozac and those popular drugs, those are all selective serotonin reuptake inhibitors. So that's how we get rid of serotonin. We reuptake it into this cell. So if we block that, it makes it build up in the synapse. Sam, do you have a question?
Yeah, what was the three ways, and one was uptake?
Enzymatically degrade, uptake into a astrocyte or a neighboring cell, or reuptake back into the neuron that released it. Okay. Oligodendrocytes, main thing here, myelinate in the central nervous system. They also provide a structural framework. If you think about all of these individual neurons running around, you need something that holds them together or in the proper place. And oligodendrocytes will do that. But remember, myelination is the main thing there. Microglia, these are immune system cells. They're phagocytes.
On the astrocytes, what does it do with the, when they absorb it? What happens with it?
The neurotransmitter.
Yeah.
Yeah, depending on what it is, it can break it down chemically and then give those chemicals back to the neuron so it can remake it. And in some cases, it will take it back and then just give it back to the neuron so it can release it again. So it kind of recycles it.
Uptake and reuptake, they both get recycled.
Right, yep. And even with, like, when you degrade it enzymatically, like acetylcholine, you get an acetyl group and a choline group, we recycle those too. Put them back together and reuse it. It's pretty efficient. Okay, so those are in the central nervous system. In the peripheral nervous system, satellite cells do pretty much everything that the astrocytes do. Regulate oxygen, neurotransmitter levels. We don't really need the microglia so much in the peripheral nervous system, because in the peripheral nervous system out in your body, you have the rest of your entire nervous system, or immune system out there. So you've got all your macrophages and antibodies, all that stuff. So you don't really need a specialized cell. And then Schwann cells, myelination, some repair. Myelination is the key thing. So you'll see, there's probably maybe 3 or 4 questions on neuroglial cells. Okay. Now we get to, let's see, this is kind of a picture of like the idea of the scaffolding and how everything's held together. Which is a pretty cool picture. So here are our cell bodies. You'll notice this is gray matter here. So we've got the tail end of myelination, but we've got all the synapses here, right? And this is white matter, so this stuff is all myelinated axons here. And here's an astrocyte. Here's our capillary. See how the astrocytes are forming a barrier around this? That's your blood-brain barrier. So stuff can or can't get through that membrane. This is some cool pictures of myelination. What's the purpose of myelinating something? Increases the speed, okay, the conduction. So in this case, the Schwann cell is myelinating. It's wrapping around there, spaced out in nodes like that, okay, super fast. And we don't get, you'll notice that in the peripheral nervous system, we don't really talk about gray matter and white matter because we don't have huge clumps of stuff. Like in the spinal cord, you have so many hundreds of millions of billions of nerves or neurons that are going in the same place, and they're all myelinated. So when you look at them, they're nice and white. You don't really have that in the peripheral nervous system because you have only specific ones going to places. So you don't have big chunks of myelinated stuff. That's kind of cool. So that's the Schwann cell that's wrapped around the axon. In this case, this is a Schwann cell also. This is not myelinating the axons, but it's just taking them where they need to go. It's giving them a route. Remember what we said about the skeletal muscle cells is that since they're electric, they need to be insulated from each other. And that's kind of the same idea here. So these axons are all going to the same place. So in this case, the Schwann cell is just acting as a conduit. Those things that'll hold on to those axons and they can go where they need to go. Okay, that's pretty cool. Damage. Okay, so let's get to the electrical charge. The membrane potential is what we're going to look at, what the resting membrane potential is, how we change it, and then the action potential. So this is our electricity. And I'm going to just draw some stuff here because I don't think these pictures are super helpful. So we start with what we call a resting membrane potential. This is the charge difference measured in voltage on the inside versus the outside of the membrane. All cells have this resting membrane potential. So if we look at our cell membrane, we want to know what this is and how or why it's the case. So our resting membrane potential is negative 70 millivolts. Okay, and it's caused by the ion differences and a couple other things. But the key thing here is that this sign tells us what the inside of the cell is relative to the outside. So if it's negative, like this, it tells us that the inside of the membrane is negative, outside of the membrane is positive. Okay? And we're not talking about, here's my cell, I'm not talking about my cell being like this big glowing negative ion. Okay? I'm talking about just what happens right across the membrane. Okay? So this is not a huge glowing difference. This is just, if this is our membrane here, I'm wondering what's the voltage in this area right here versus this area right here, okay? So it's a little bit negative on this side and positive on this side. Well, how negative is it? It's 70 millivolts negative. Why is it 70? It's just how it is, right? It could be a million, it could be 100, it could be anything. It's just negative 70. It's just the way the cells are. So how do we do this? Or why? Why is it negative? And it's going to be this membrane potential here that we're going to manipulate in an action potential. Okay? So some cells, if you're a cell that has electrical excitability, like neurons and muscle cells, you have the ability to change this and manipulate it. Okay? And that's our electrical signal that we're going to move. But how does this work? So there's a couple things here. Here's my cell. Here's my sodium-potassium pump. How does sodium-potassium pump work? Here's the outside. Here's the inside. How does my sodium-potassium pump work?
Three.
Other way around. We move 3 sodium out and 2 potassium in. So in that activity, which all cells do, there's a net movement of positive things out of the cell, right? We're moving more positive things out. That's making the inside of the cell negative. Okay? Imagine that I've got a situation like this, where I have 10 negative things and 10 negative things on each side. Okay? I move 3 positive things this way and only 2 positive things this way. This side's more positive because I'm moving positive things that way. Okay? So this is one of the things that leads to my negative inside of the cell. Okay? I'm moving more positive things out. Now both of these can leak back in. We have leak channels, ion channels that are just open. Ions just move through them. If sodium is allowed to leak or move, which way will it move? Always. If you let sodium go where it wants to go through diffusion, where will it go? Down its gradient into the cell, right? So that's important. If we open if we have any sort of a sodium channel anywhere, anytime, any place ever you see it, sodium moves into the cell, always down its gradient, okay? Sodium concentration is always higher on the outside of the cell. It's always true. What happens if you open a potassium channel? Potassium concentration is always, always greater on the inside of the cell. So potassium will always go out. So if we open a potassium channel, potassium moves out all the time. And this, knowing this right here, this is everything that we're going to do to manipulate this and get our action potential. Okay, so you have to know this. So first thing I'm doing is I'm moving more positive things out with my sodium potassium pump. Okay. Now I'm going to let sodium and potassium leak down their gradients, and that's also going to produce a net movement of positive things out of the cell. So the question is, what is the cell more? What leaks more, sodium or potassium? What's the cell more leaky to if the net movement is positive things out? Potassium, right? If you open a potassium channel, potassium goes out of the cell. So here's my cell. Okay. I've got these little channels in here, and these channels are always specific for an ion. So I've got a sodium one here, and I've got a potassium one here. I want to have a net movement of positive things out of the cell. So that means more potassium moves out than sodium moves in. Okay? So the cell is more leaky to potassium. That's the second thing that causes this negative. So more potassium leaks out than sodium leaks in. Okay? So those two things, both of those, I have a net movement of positive things out, makes my cell negative. The third thing that makes it negative is that inside my cell I have some proteins that are anionic. I have some negatively charged proteins. Inside the cell. Okay. So those are my three things. So every cell in your body is doing this. Little keratinocyte in your epidermis is doing this, right? Neurons, muscle cells, cells in your liver, cells in your kidney, you name it, it's doing this all the time, every day, 24/7. Okay? Tons of ATP to do this. Cells always leaking more potassium out. Cells always got negative proteins inside it. So we always have a resting membrane potential of negative 70 millivolts. Okay? If you are an electrically excitable cell, like our muscles and neurons, you can manipulate and change the resting membrane potential. Okay? If you don't have electrical excitability, you can't do that. You can't do anything about it. It just sits there. So let's talk about what we're gonna do to manipulate this is we're gonna open ion channels. And all ions have their own channels. Okay? So we have potassium channels, we have sodium channels. What's another ion channel we've seen? Calcium. And we have chloride ion channels. These are the main two we're going to look at. And then we're going to toss this one on. This one, these channels aren't really involved in the electrical part of this. They're opened and closed by the electrical part of this, but they're not involved in making the electricity. So my channel types, I have leak channels that are just open. So imagine a membrane, it just has a little channel in there and the ions can just leak through it down there gradient. Okay. And usually in my equilibrium, I've got more potassium inside, I've got more sodium outside. Chloride always hangs out with potassium or sodium, so it's outside. And usually I have more calcium outside also. Okay. So, like we said over here, if we open a sodium channel, sodium will move into the cell. If I open a potassium channel, potassium will move out. What happens if I open a chloride ion channel? Where will it go? In, down its gradient, right? High concentration outside. So if I let it flow, it's going to come in. Calcium will do the same thing, okay? Which we saw at the end of the axon, right? Where calcium comes in. So I can have leak channels for these. And those are just random channels. They don't have a trap door on them. They're just open. Ions just leak through them. Right? Remember that these channels are proteins. And the cell determines which proteins that they make. So it's not like we say that a cell is leaky to sodium or potassium and it can't do anything about it. If it's leaky to one of these ions, it's doing it on purpose. It's making that protein and putting it there, okay? So that's one type of channel. What's another type of ion channel we've seen? A gated channel. There's a couple gated channels. What's that? Chemically gated is one. What's the one that opens the calcium channel? Voltage gated. Okay, so these are just open. And the others we have are gated. They got trapdoors on them. Okay, so we also have voltage gated channels. These open or close when the membrane voltage changes. Okay. So we saw these in the sarcoplasmic reticulum, right? And that was the whole purpose of the electrical signal that came from the neuron, right? Go back to your muscle cell. So release acetylcholine, we start that action potential or that electrical charge on the sarcolemma, down the T-tubule, past the sarcoplasmic reticulum. We did that to open these channels because they open and close when the membrane voltage changes. Okay? And they can open and close at different voltages. But that's how it's going to help us manipulate this too. Another type is a chemical or receptor mediated. So these open and close when something binds to a receptor, whatever that something is. This something that we're gonna see usually is a neurotransmitter or maybe a hormone. And the last type is called a mechanically gated. These open and close when the cell is manipulated physically. So here's my membrane. Here's all my channels. Okay, 4 types. First type is just open. Just sits there. It's a leak channel. No trapdoor, it's just there. All cells have it. Okay. Cells have more potassium leak channels than sodium. That's why more potassium leaks out. Second type, here, has a trapdoor on it. Trapdoor is going to open and close when I change the voltage. What's the resting voltage? -70, right? If I can manipulate that and change it, I can open that gate, open that channel up. That's a voltage gated channel. So if I've got a way to move those ions around, and let's say I can make this a +30, that can open a channel. Okay? So all I have to do is change this number, change this voltage, and I open that channel. Okay? Voltage gated channel. This one, I need a receptor for, the third type. So now it's got a trapdoor, and that trapdoor opens when something binds to the receptor, like a neurotransmitter opens up. Did I draw too many? I did. My fourth type, and this is a weird one. So imagine in the skin, I've got these special nerve endings like this. So this is my skin, and this is that special receptor of a sensory neuron. I press it, I poke it, so I physically manipulate the membrane. That's what opens that channel, is physical distortion of that cell. Okay, we really find that only in receptors, and that's how all pressure and touch works. When you manipulate that little cell, squeeze that little cell, it opens the trapdoor. Okay? So those are our types of ion channels. These are always around. These are the ones we're going to use to get our action potential, our electrical signal on the membrane. Okay? These are the ones we're going to use when we want to send a signal to the next cell using a neurotransmitter. And these are the ones that we're going to find only in a couple cases, and we're not really going to worry too much about. Okay. So now, that's our resting membrane potential. Now we talk about changing it. Okay. The only cells that can change this are special cells. Change it. So let's draw something like this. And this is, if you flip the page, let's see. Oh, that next page has the different types of channels in it. Chemically gated, voltage gated, and mechanically gated. Okay. And then the next picture, this one, this is the one where we start to talk about changing that membrane potential. So here's my -70. resting membrane potential. If I don't change that cell or do anything to it, that voltage just stays exactly like it is. If I come in here with some sort of a stimulus, and I stimulate that cell, we'll talk about what that means later. If I stimulate that cell somehow, and I can open sodium channels, what happens? So if I open sodium, let's think about it this way. I'm at -70. I open a sodium channel. Which way will sodium move? Into the cell. So sodium comes in. What's that doing to this number? It's making it more positive, right? So I'm at a -70 right now. If I'm adding sodium, because I'm opening the sodium channel, this number becomes more positive. I'm changing that membrane potential, okay? So if I give a little stimulus here, and I open a couple sodium channels, a little bit of sodium rushes in, and this goes up. Okay, let's make it look like a nice curve. Okay, and this number goes up a little bit. If I stop that stimulus, all those channels close. Sodium gets pumped back out, and this goes back down to normal. Let's say I've got up here to a -68. I've made it a little bit more positive. Anytime I make the membrane potential more positive, it's called a depolarization. And the only way I can make it more positive or depolarized is to open sodium channels. And anytime I open a sodium channel, it will depolarize that cell. It will make it more positive. So now I'm at -70. And I open potassium channels. So here I am, I'm at a -70. What happens when I open a potassium channel? Where does potassium go? Goes out, right? It's always higher inside the cell. So if I open a potassium always go out. So now I'm removing positive things. And this number is becoming more negative. -71, -72. Because I'm taking positive things away. If I do that, when I drop down here, like this, and then I come back, when I go below resting membrane potential, that's called a hyperpolarization. So if it's more negative than resting membrane potential, hyperpolarization. If I open potassium channels, I can do this. Okay. Where's there more chloride ions? Inside or outside? Outside. So what happens if I open those channels? Chloride flows in, and chloride is negatively charged, so it makes the cell more negative. So I can also do this if I open chloride ion channels. Okay. So all I'm doing is I'm moving positive or negative things to make a membrane more positive or more negative. That's it. That's all I'm doing. If I do this just a little bit, like this, and I don't get a great big change, and it just kind of goes back to normal, those are referred to as graded potentials. It's a partial change, but it stops. So a partial change is a graded potential. big this change is gonna be depends on how strong this stimulus is in the beginning. So it turns out that in electrically excitable membranes, there's a voltage up here that is called a threshold voltage, which means that if I get to that level, then I get an action potential. Let me say this. Action potential is a rapid reversal in membrane potential. It's so rapid and so big, when I get this, I'm gonna go from -70 all the way up to +30. I'm gonna change that membrane potential completely. That's the electrical signal we've been talking about all this time, is that voltage change from a -70 to a +30. The threshold number to get an action potential, your book says -60 millivolts. Other books will say -65 or 55, doesn't matter. So what we're saying is that if I can give a stimulus here that can open sodium channels, enough of them to get up to -60, like this, that's the threshold. I do something special and sodium rushes in and I go all the way up to a +30. Okay? If I don't get to -60, I come back down and have a graded potential. So the threshold is an all or nothing deal. Either I get an action potential because I reach that, or I don't. Okay? We'll talk about how we reach that.
Okay, so if it gets to -60, that's enough for it
Yep.
to create that action potential.
Correct.
And if it passes it, then what?
Well, once it reaches it, it's gonna explode,
Okay.
and you're gonna get an
So it's
action
not
potential.
relevant that it goes up to +30.
It is, but we'll talk about why that happens. To get up to +30, you have to get to -60,
Okay.
okay? And +30 is our action potential. That's the change that we need. Okay. So, imagine that. Here's my membrane, okay? And I'm talking about this little section of it here, okay? Little teeny weeny section. I've got all these sodium channels here. Let's say I open one sodium channel. A little bit of sodium flows in, right? My number starts to go up a little bit. So I've opened one, so I start to depolarize a little bit. If I stop right there, I've only gone up a little bit, comes back down. Let's say I open another one. So now I open two. A little more sodium comes in. I'm getting a little bit higher, right? The number of these that I'm gonna open depends on my initial stimulus. And we'll talk about that when we get to it, okay? So I'm stimulating this cell somehow, and that stimulation is causing sodium channels to open, okay? If I keep stimulating this cell, I keep stimulating it, I keep opening more and more of them. Every sodium channel I open, more sodium flows in, right? So every sodium channel that I open, I start to depolarize my membrane a little bit more. So the idea is, if my initial stimulus is strong enough to open enough sodium channels to get this up to -60, that's the number I need where all of the sodium channels in this area open. Because they're voltage gated channels. And the voltage that they all need to open is -60. So here's a voltage gated sodium channel. It's just sitting there, it's waiting. -70, not gonna open. Negative whatever, 68, not gonna open. -65, not gonna open. -60, boom, open. That's the stimulus that causes it to open, okay? So I'm giving some other sort of a stimulus here that's causing them to open a little bit. Once I get to -60, all the voltage gated channels open and sodium floods into the cell. And that makes this number shoot up to positive 30. That's the first step of an action potential. Okay? So if you look at the next picture, you see the phases of an action potential. And that is the depolarizing phase. That's called depolarization. Phase 1. And these phases of action potential you'll want to know for the exam for sure. Okay. So that's my rapid reversal. So here's my membrane. It's long. Here's my cell body. Here's where I want to release my neurotransmitter, way down here. Right? If I want to release my neurotransmitter down here, what type of a channel here do I have to open? Voltage-gated calcium channel. So I've got a calcium channel sitting here, and it's going to open when I change the voltage on the membrane, right? Well, I just changed the voltage on the membrane back here, way back here, this little teeny weeny section. So if I look at the rest of this cell, it's still normal like this. Okay? But what I've done right here, just in this little teeny area, is I've opened enough sodium channels to depolarize it. So now I've changed it. Now the cell is a positive 30. That's my rapid reversal in membrane potential. Okay? That's the number I need to get down here to open that channel. So this is a voltage-gated channel too, right? It's sitting there. What's the voltage? Negative 70, not going to open. Negative 60, not going to open. Positive 30, poof, open. Calcium comes in, neurotransmitters get released. Okay? So I've changed the membrane potential down here by opening my sodium channels. And now what I have to do is get this change in membrane potential down there. So I can open calcium and release my neurotransmitters. That's the whole reason I'm doing all this. Okay? So I've just depolarized my cell. And that's great. But now I have to do two things. I have to move that change in membrane potential down the length of the axon, right? So I've got to propagate it. But I've thrown this part of the cell way out of homeostasis. So if I want to send another signal, I've got to switch this back really fast. So the second part of my action potential is to get it back to normal. So now I'm up here, and I want to get it back down to negative 70 as fast as I can. So here, I want to close my sodium channels. Okay? Negative 60 was the number that opened them all. Positive 30 makes them all close. Okay? So now the sodium-potassium pump can work and pump sodium back out. So what I've done is, in this area, I've brought a ton of sodium in. Now I've got to get it out. If I let the sodium-potassium pump work, 3 sodium at a time, it'll work, but it's going to take a long time, right? If I did that, this would go back down, but it would go down like this, really slowly, as I slowly pump that sodium out. Right? I don't want to do that, it's too slow. So what can I do now to bring this back down really fast? How can I make the cell more negative? What can I open? Potassium channels. So I've got these voltage-gated potassium channels sitting there also, right? Negative 60, they don't open. What do you know? They open at positive 30. So when I hit positive 30, I close these, but I open my potassium channels. And now potassium flows out. Flows out. That drops this down. That's repolarization. Back down to resting membrane. and I overshoot the mark a little bit, so I go below it. And then the sodium potassium pump brings me back to normal. So this is the second phase. The third here is hyper. The reason that we do that is not significant for you guys. Okay. So what I've done in this little section of my membrane here, just in this little teeny weeny section, I've opened my sodium channels to let sodium flow in, get to positive 30. Then I open my potassium channels and let potassium flow out, get back to my negative 70. Okay. So I have this rapid reversal in membrane potential, and then I get back to normal. That's the action potential. That's the electricity. Okay. And I'm only doing it by moving positive things. So it's kind of, in one way, it's a really simple idea because I'm just moving positive things and thinking about the result, the numbers, right, the charge. But it's weird that we're doing it this way. Okay. So now that I've done that, now I have to move it. My goal is to get it down here so I can open my calcium channels. So now that I've done it here, I gotta move it. Okay. So what the deal is that the rest of my membrane is normal, like this, right? Negative 70 here. So think about what's happening initially with my sodium. So when my sodium starts to flow in, like this, it's got to go somewhere, right? It's positive. What's it attracted to? Negative. So when these sodium ions start to come in, they see this positive part of the membrane and they start to migrate over here because they're positive. What does that do? That starts to open voltage-gated sodium channels here. So now sodium starts to flow in here. And more sodium starts to flow in here. So I reach threshold here. All this sodium floods in, moves over here. I reach threshold here, sodium floods in. Same thing happens. All this sodium comes in, moves over here. So if I watch the movement of my action potential down the membrane, I would see sodium coming in here and migrating over here. Sodium coming in here and migrating over here, like this. That's how I propagate my action potential. Okay, that sodium floods in and then moves over to the negative part. And as soon as it moves over here, so this at rest is at negative 70, but sodium's coming over here. It's making it more positive until eventually this area gets to threshold. And now all the sodium flows in here, migrates over. This area reaches threshold. Sodium flows in here, migrates over. Okay. If I do this all the way along here, it takes a long time. That's continuous propagation. So this is an unmyelinated axon. I start my action potential here, and it goes stepwise, like this. It's called continuous propagation. Slow. If I myelinate my axon, like this, now what's happening is my sodium's flowing in here, just like it did before, but the negative part of the membrane that it can get to is way over here. So that sodium flows in here and skips over here and starts an action potential. It skips over here, skips over here, skips over here. So when I myelinate my axon, I let this change in membrane potential skip from one node to the next. That's why it's faster. Okay? It's still the same thing, but and evolution has determined how far this should be, right? If I have a myelinating cell that's this long, and my sodium comes in here, it's too far. I couldn't get it all the way over here, wouldn't work, right? If I make these things too short, then it'll speed up my action potential, but it won't speed it up enough. So you'll find that the length from node to node is an exact space that evolution has determined to make it the absolute fastest it can be. Because if it was further, it wouldn't work. If it was shorter, it wouldn't be as fast. Okay? So this is myelination. And this is called saltatory conduction or propagation. Okay, one last thing to tie some of this together. So this is a head scratcher. Here's my multipolar neuron, okay? I've got all my voltage gated channels on here, just sitting there, resting membrane potential, everything's normal. Another neuron comes in here, releases neurotransmitter there. So now if I blow that membrane up like this, I get something like this. Here's my dendrite down there. Here's my neuron releasing neurotransmitter. This is a chemically gated or receptor gated ion channel. So now let's say this is a sodium channel. It's got a receptor on it. My receptor binds, or the neurotransmitter binds to this, opens the trap door. What happens? Where does sodium go? Into the cell. What does that do to my membrane potential? I start to get an action potential, okay? So when we talked about the stimulus at the beginning, this is the stimulus. How many of these am I opening based on how many things bind to the receptor? So if this neuron only releases a couple neurotransmitter molecules, then I'm only going to open a couple of these. And this number is only going to go up a little bit. If I release 1 million neurotransmitter molecules, then I can open 1 million of these, and I can get this number up really fast. Okay. What happens if, instead of a sodium channel, this is a potassium channel? Now what happens to this cell? I open this up, potassium goes out, this hyperpolarizes, right? And nothing happens. So now I've got all of these things here, like this. And remember that the presynaptic cell can determine the number of neurotransmitters it releases, right? But the postsynaptic cell determines what type of receptors it has. So if this cell really wants to be stimulated for some reason, it's going to make a whole bunch of sodium channels there. Gets really stimulated. If it doesn't, if it's lazy, wants to take a nap, it can make none at all, or it can even make potassium ones. So not only does it not get stimulated, it gets inhibited. So now imagine in this cell, this one's opening a sodium channel, this one's opening a potassium channel, just at random. This one's sodium, this one's opening a chloride channel, potassium, blah, 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 blah. They all have different effects, right? So this is going to depolarize the cell. This is going to hyperpolarize the cell, all this stuff. So what we have to do is take all the effects of this and determine what happens here. That's the axon hillock. So that's when we said the axon hillock is where the decision is made. Is this neuron going to send an action potential or not? It depends on if we reach threshold at the axon hillock. And that's determined by all the effects of this. Okay. So if this cell really super wants to be stimulated fast, it can make all of these sodium channels. And then you reach threshold here instantly, and there goes your signal. Okay? If this cell wants to be inhibited and not be active, it just makes all these potassium channels, and it doesn't do anything. So that's when we started here, and we drew this picture, and we said, here's the initial stimulus that raises that up a little bit. That initial stimulus is the number of those receptor-gated channels we open. Okay? So that's determined by how many of them there are and how much neurotransmitter is released. Okay? So some of them will stimulate the cell and some of them won't. Okay? So that's the significance of the axon hillock. That's the decision-making point. Okay? That's how it all works. So I move that down here, open my voltage-gated calcium channels, calcium comes in, release my neurotransmitter. And then whatever's gonna happen to the next cell is up to that next cell. That's it, that's how they all work. That's how all the neurons work. That's how muscle cell works too. So when we said the sarcolemma, down the T-tubule, that's what's happening. That change in membrane potential, -70 to +30. It's the exact same thing, okay? So remember, action potential is an all or none event. Either you reach -60 and you get it, or you don't. You can't have a partial action potential, okay? All right. So then, on Monday, we get into the brain, and what you guys will notice is that I put a lot more information in this outline than I did in some of the others, which is why it's so long. Because for a lot of the stuff with the brain, you have to identify where it is in lab, but in lecture, we just need to know the function of it. So a lot of that I put in here for you. So for a lot of what we do with the brain and the spinal cord, you won't have a lot of notes to add. Because why should you have to write down posterior tip of the occipital lobe? I just put it in there for you, okay? So read through the part on the stuff on the brain, try to get a hang of this action potential, okay? You're definitely gonna see questions about ion channels, graded potentials, action potential, propagation, myelination, all that stuff will be on your exam, okay? All right, guys.